Roxana presented herself yesterday very well. So she's a computational neuroscientist, and she's interested in mainly in how timescale can be in the dynamic of the brain dynamic activity is related to behavior, and that's what she will talk about.
Well, thank you so much for the organizers for organizing such a wonderful workshop and also giving me the opportunity to share some of our work with you. Today, I'm going to talk about one of the important features of our behavior, which is we humans and other animals can survive in the environment because we can adopt our behavior very flexibly to different situations that are happening around us. And one of the hallmarks of this adaptivity is being able to perform behaviors and computations over a broad range of timescales. For instance, our brain can perform very fast reactions for us to survive in the environment. And at the same time, we can process information over slower timescales. For instance, in the case of this cheetah, it can accumulate information over long timescales in order for this cheetah to make a decision whether or not to attack the deers. Parallel to this broad range of timescales in the behavior, we also have a broad range of timescales in the fluctuations of neural activity in our brain. For instance, here we are looking at firing rates of some example neurons, and you might have noticed that some of these neurons fluctuate over relatively slow timescales, and some have much faster fluctuations in their activity. Growing evidence suggests that these different timescales of neural activity fluctuations might actually be related to the behavioral timescales. For instance, if we look at the timescales of neural activity fluctuations in sensory areas, such as primary visual cortex, we see that neurons in these areas have relatively fast fluctuations, which could be relevant, for example, for tracking fast sensory inputs. At the same time, if we look at the fluctuations of activity in higher cortical areas, such as prefrontal cortex, we see much slower fluctuations in the activity, potentially relevant, for example, for performing more cognitive demanding tasks, such as decision-making or working memory. At the same time, it has been suggested that these timescales are not fixed properties of our brain, but they can adapt to the task requirements. For instance, engagement in a working memory task can increase the timescales of neural activity fluctuations. Today, I'm going to talk about, again, the adaptivity of timescales, and I will try to answer the two following questions. So first of all, what is the relevance of adaptive neural timescales to behavior and brain computation? And second, what are the neural circuit mechanisms that could potentially give rise to this adaptivity in our brain? We tried to answer these questions from two complementary but different perspectives. And during this talk, then we're going to look at these two different things. So in the first part of the talk, we're going to look at the timescales of neural activity recorded from monkey visual cortex. And we want to understand how these timescales would relate to the behavioral state of the animal. In the second part of the talk, we're going to look at the timescales of neural activity from artificial recurrent neural networks, and we want to understand how these timescales will be relevant for the types of computations that these networks are trained to perform. So now let's start with the first part. So here we want to understand, do neural timescales in primate visual cortex adapt to the behavioral state of the animal, which in this case, it is defined by the attention state of the animal? And if they do, then what are the neural circuit mechanisms? So to answer these questions, we analyzed spiking activity recorded from primate visual cortex while they were performing different types of selective attention tasks. For instance, during one of these tasks, the monkeys presented with 4 different visual grating stimuli. One of these stimuli is located at the receptive field of the neurons that we record from. So the neurons that we record from would only see this part of the screen. The task of the monkey is to report at the end of each trial whether the orientation of any of these grating has changed. And it is presented with an attention cue during each trial that would guide the attention of monkey to the location that is most likely the place that this change of orientation would happen. Then if the monkey thinks that there is a change of orientation, for example, in this part of the screen, then they need to make a saccade to the opposite location. So there is an anti-saccade response. And because of the anti-saccade response, then 2 of the stimuli are the target of attention in condition, which means that the monkey is either covertly or overtly attending toward the receptive field of the neurons that we record from. And the two other stimuli, are the target of attention away condition. So we don't expect any attentional modulations in those cases. And because of this, then we also have two different behavioral states. So we have attention in state, so the trials that we expect to see attentional modulations of the neural activity in the part of the brain that we're recording from, and attention away, which serves as a control condition. Our collaborators in Terry Moore's lab and Alexander Thiele's lab recorded spiking activity from primate visual area V4 using these types of laminar microelectrode arrays. And it will allow us to measure spiking activity across different cortical layers simultaneously. So this is one example session of this recording. So we wanted to know how timescales of this neural activity would change across these two different attention conditions. And for this, what people usually do is to compute spike count autocorrelation, which is just a very simple measure. So we just bin the spiking activity to get a time series like this one, and then we compute the autocorrelation function of the spike count, which is just shifted version of the time. So computing the covariance between the original, the shifted version, and then normalizing it by the variance. So super simple measurement. Usually when we look at these autocorrelations, they look like this exponential decay function. So how fast this autocorrelation decays over time give us an estimation of the timescales of dynamics. Instead of plotting these autocorrelations in linear coordinates, we can also plot them in logarithmic linear coordinates. And this is a very useful representation because there we expect that each exponential function should appear as one straight line, and the slope of the line gives us the time scale. So if we have more than one time scale in the data, then we expect to have multiple lines with different slopes. And this way we can identify potentially how many time scales we have in the data. So let's together now look at some example autocorrelations from our data. So this is autocorrelation of population activity from an example session across two different attention conditions plotted in logarithmic linear scale. And the first thing you might have noticed by just looking at this autocorrelation shapes is that there are multiple slopes in this autocorrelation. It's not just a single exponential decay. So most likely there is a faster time scale at the beginning, and then there is a slower time scale in later time lags. And it looks like, at least in this specific example, the time scale, the slow time scale is increasing during attention. So we can just then make two hypotheses. There are at least two time scale, the fast one and a slow one in V4 ongoing dynamics, and maybe the slow time scales are modulated during attention. However, to test this hypothesis, we need to precisely measure these time scales from all the data and then start comparing them. Usually what people do to measure time scales from autocorrelations is to fit an exponential or a mixture of exponential function to the autocorrelation function, and then use this parameter tau from the exponent as the estimation of time scale, which makes sense because usually when we look at these autocorrelations, at least in linear domain, they look like this exponential decays. However, this method does not always give us reliable estimates because the shape of data autocorrelation pretty much depends on the statistics of the data, including the length of the trials that we were recording. And also in the case of spiking activity, for example, the firing rates. To understand what the problem is, let's for a few minutes together forget about neural data and just look at some synthetic data that we know what should be the ground truth shape of autocorrelation and the ground truth timescales. For instance, we can consider Ornstein-Uhlenbeck process, which is a simple stochastic process. We know analytically the autocorrelation shape is an exponential decay function. We can set its time scale by hand, so there will be a parameter tau. So we expect the ground truth shape of this the autocorrelation of this process to be just this gray line with the given slope in the logarithmic axis. Now I'm simulating this process and I get data from different trial durations. For example, let's say for 1 second and 4 seconds. And I'm plotting the autocorrelations of this data here. So the first thing you notice is that, first of all, the shape of these autocorrelations are not just a straight line. So you see just there is a bend here. And despite that these data, so these autocorrelations are measured from the same data with exactly the same time scale, they actually look very different depending on how long was our trial duration. And if I were to estimate the timescales by just simply fitting an exponential function, so it will be like the straight lines, then depending on the length of the data I had, I would actually underestimate my timescales. So this is a known statistical problem in statistics. It's called bias in sample autocorrelation, which suggests that the shape of data autocorrelation alone is not a good representative for measuring timescales. So to overcome this problem, we developed a method, a Bayesian method, that uses generative models to estimate timescales instead of fitting directly the autocorrelation shapes. I'm not going to go through the technical details of the method, but I'm more than happy to talk about it if someone is interested afterwards. But the main idea is very simple, actually. So imagine that we recorded data from the brain with some like given trial durations, and we are able to measure simple statistics. For example, we can measure firing rates, we can measure variance, we can also measure autocorrelations. Now, if we can come up with a generative model that can produce synthetic data with the same statistics as our real data, so for example, if it can create data with exact same shape of autocorrelation, even though it is distorted, then we can use the ground truth timescales of the generative model as the estimated timescales of our real data. So to fit the generative model to the real data, we are using a method called approximate Bayesian computation, which at the end of the day would return a posterior distribution of timescale. So instead of telling us, for example, the timescale of this data is 80 milliseconds, it would tell us the timescale is somewhere between maybe 60 to 120 milliseconds with a given probability distribution. And for generative models, we are pretty flexible. So we can use different types of stochastic processes depending on the data that we are modeling. For example, at its base, we can use a mixture of orthogonal processes. And for spiking activity, we can use different types of renewal processes to generate spikes. We can also use the same method to identify how many timescales we have in the data. For instance, I can fit the data with a model that only has one timescale, a model that has two different timescales, and then use Bayesian model comparison to find the best model and the best number of timescales that would describe our data. So developing this model, then we wanted to answer two questions. One is that how many timescales we have in our data and whether they are changing during attention or not. So first, we fitted all the data autocorrelation across different conditions with two models, so a generative model that only has one timescale and a two timescale model. And here you can see that for the majority of our data, when we do the model comparison, they are better described by the two timescale model. We'll also perform similar analysis to compare between 2 and 3 timescales, and there we also found 2 timescales, at least for our data, was a better description. If you're also curious about the range of these timescales, so our fast timescale is relatively short, so it's around like tens of milliseconds, and the slow timescale goes like up to 200, 400 milliseconds. So now let's go back and look at the same example that we saw a few slides ago. So now that we know that the autocorrelations are better described by 2 timescales, so we are fitting every autocorrelation with the model that has 2 timescales. And we want to understand if there is a change in that during attention in the timescale. So here in this example, I think this is what's even clear from the beginning, that there is no change in the fast timescale. So these are the marginal posterior distributions for these 2 autocorrelations. But the slow timescale is increasing during attention. But if you look at like data by eye, it is not actually very clear for all the sessions that this exists. But using our method, we could actually show that this is true for the rest of our data. So another interesting thing that we found was that what is the relationship between the timescales of neural activity and the behavioral timescale of the animal. So in this case, we are looking at the reaction times of the monkey to the change of orientation at the end of each trial. And we could find that there is some sort of negative correlation between the slow timescales and the reaction times of the monkey during attention in condition, but no such correlations during attention away condition, suggesting that maybe there is some like relationship or some behavioral relevance to this attentional modulation of slow timescales during this task. So far we have seen that we have at least two different timescales in V4 ongoing activity, and it looks like the slow timescales is kind of behavior relevant. So it was increasing during attention, had some correlations with the reaction times. The question is that how these timescales are now generated in the primate visual cortex. There is a large body of literature actually looking into different types of mechanisms that can create different timescales in our brain. And we can think of them across two separate groups. So the first group would relate the timescales of neural activity to the biophysical properties of individual neurons. So we can think of it, for instance, as membrane time constants, synaptic time constants, and things like this. The second group of mechanisms rely on network interactions to generate timescales, and they would suggest that both the strength and the structure of the connectivity would give rise to different timescales in the neural activity. So based on these ideas, we also came up with two different types of models to propose how these two different timescales, so fast and slow timescales, are created in the monkey brain. The first group of models assume that these timescales are only coming from the biophysical properties of individual neurons. For instance, we can build a model with a fast membrane time constant and a slow synaptic time constant, and then each neuron autocorrelation would have this two timescale autocorrelation shape, similar to what we saw in the data. The second group of model would connect this slow timescales to the recurrent interactions in the network. So in this model, the fast timescale still comes from the properties of individual neurons. So we can think of it as a biophysical time constant. But the slow timescale is created through local recurrent interactions in our model. And specifically, we built this model to have this local spatial connectivity mimicking the primate visual cortex connectivity structure. So each neuron would be connected to its nearest neighbors within spatial radius. And then having this local spatial connectivity, then we can show that, again, neurons would have two timescales in their activity. Here, the fast one is coming from biophysical properties, and the slow one comes from the network interaction. So now we have two different models with completely different mechanisms, both equally well explaining our data. So the question is that, is there a way to distinguish between them? And it turned out that if, in addition to the autocorrelations, we look at the cross-correlations between the activity and how the timescales of those cross-correlations depend on the physical distance between the neurons, these models could actually have different predictions. So in the first model, we assume timescales are only coming from biophysical properties of individual neurons. Therefore, we can assume that network interactions do not play any role, and we can assume that the connectivity is random in these networks. So if in a random network, I compute the cross-correlation between the activity of each two neurons and see how those cross-correlations depend on the distance between the neurons, because in a random network, there is no meaning of a physical distance, then there is also no dependence on the distance. So this is just a trivial result. However, in the spatially connected networks, if we look at how the cross-correlations between the neurons change with the distance, first of all, we see that as the distance between the neurons increases, the strength of these cross-correlation would decrease. But the more interesting result is that they become also dominated by slower timescales, which means that neurons that are further away from each other are correlated with each other over slower timescales compared to the neurons that are closer to each other. So now we have a prediction that we can go and look in our data and see which one is more better descriptive of our data. So we use the same Bayesian method to measure timescales from cross-correlations, and we could show that we see an increase in the timescales of cross-correlations in the data as the distance between the neurons is increasing, which would support the predictions of the spatial connectivity model. So then we use the same model to also ask how the attentional modulations of the timescales is created in our brain. And for this specific model, we can also compute the timescales analytically, and we can show that the fast timescale only depends on the intrinsic properties of individual neurons, whereas the slow timescales would depend both on the fast timescale, but also the recurrent interactions. Now, the interesting thing is that if in these models we introduce nonlinearity in the recurrent interactions, we can show that additionally external input to the network could also modulate the slow timescales through changing the effective recurrent interactions in the network. And this could be one potential explanation for attentional modulations of the timescales, because we know during attention we have top-down attentional inputs from higher cortical areas to V4, and they could potentially be responsible for changing the slow timescales through the local spatial connectivity that exists in the prime brain. For those of you who are interested in criticality, we could also show that the parameters of the spatial network model that would match the different attention condition would suggest that during attention, the model is becoming closer to the critical point. So this might be also relevant, for example, if we think about, like, if during attention, we do need like a more efficient code of the stimulus that we are seeing. So maybe being closer to the critical point is an actual relevant property. And this is like a different line of research that has been shown in our lab, that indeed networks that do efficient coding are also close to the critical point. So maybe there is also some interesting thing happening there that during attention, we can also do better computations when we are closer to the critical point. So far we have seen that the slow timescales adapt to the behavioral state of the animal. And if we have to choose between a model that only relies on biophysical properties and a model that only relies on network interactions to describe how these timescales are generated, we need to choose the model with network recurrent interactions to explain the timescales. However, in our brain, both of these mechanisms exist at the same time, so we cannot choose one or the other. So the question is that if both of them exist at the same time, and if we need to learn tasks that require adaptive timescales, which one is better to optimize? Should I optimize the properties of my individual neurons to develop adaptive timescales, or should I optimize the recurrent interactions? And this is precisely the question we want to answer in the second part of the talk. So we want to understand which of these two types of mechanisms is optimal for learning tasks that require adaptive timescales. So in this part of the talk, we are going to use artificial recurrent neural networks, or RNNs for short, as a kind of model to study the relationship between timescales and computation. As you probably know, RNNs are a good model because they can mimic the recurrent interactions in our brain, and they can generate like interesting dynamics. But at the same time, we can use machine learning methods to optimize them to perform different types of tasks. So they would give us a very nice handle to relate neural dynamics directly to neural computation. And then by reverse engineering those RNNs, then we could understand potentially what types of mechanisms could give rise to the computations that they are performing. So in our study, we are training our RNNs to perform different types of n-back memory tasks. In each task, the network will receive a binary input over time, and it will produce a binary output over time. And each input digit is presented to the network for a finite duration of t. In one of these tasks, for instance, in the n parity task, the network needs to compute the binary sum of the last n digits at every moment of time and then report it at the output. So it is an online kind of n parity task. And then in this task, n defines the memory requirement of the task. So the larger the n, the more digits I need to keep in my memory. And because of this, then we expect that for solving a task that has a larger n, we need to have slower timescales in the dynamics in order for these neurons to be able to integrate information over longer timescales. But the question is that how these slow timescales are generated in these networks. Are they coming from properties of individual neurons, or would they come from network interactions? And we designed our model such that both of these parameters are trainable throughout the training, so network can actually optimize either one to learn the task. The dynamics of each neuron in our network is given by this equation, so it's somewhat similar to a leaky integrate fire neuron. But for the rest of the talk, I only want you to remember two parameters from this equation. One is this Wij parameter, which is just a recurrent connectivity matrix in our model. Another one is this tau time constant parameter. So for each neuron, we also define a time constant parameter, which is trainable, and it defines the time scale of the neuron in the absence of any network interaction. So we can think of it as an approximation of a biophysical time constant. In addition to this time constant parameter, we also define an effective time scale, which we measure directly from the neural activity autocorrelation of each neuron. And this effective time scale potentially is dependent both on the recurrent connectivity, but also the time constant. So it represents the time scale of the neuron when it is embedded in the network. Now, if we try to follow how both this tau and tau effective are changing throughout training, we might be able to, at the end of the day, say which one was more important for learning tasks. So would neurons rely on the time constant parameters to adapt to the memory requirement of the task, or would they rely more maybe on recurrent connectivity? So to answer this question, then we need to train our RNNs to perform relatively long memory tasks. However, this is a problem in machine learning. RNNs are not very good at learning long memory tasks. To solve this problem and help our networks to learn long memory tasks, then we turn to curriculum learning. And when I say curriculum, maybe some of you are already thinking about the school curriculum, and we also had this interesting talk about the curriculum yesterday. And I think that's the right analogy here. So when in machine learning, we think about curriculum learning, we're also thinking about two concepts which are very similar to the school curriculum. One is gradual learning of a difficult task. For instance, in the school, the things we learn in the first grade would help us to learn the things in the second grade better. And we also have this concept in machine learning. We also think about compositionality. So the tasks that we learn together might actually help us learn them better. For instance, if we learn maybe physics and math together, we might be able to understand physics better. So with these ideas in mind, we also came up with two different curricula to help our RNNs learn this long memory task. In the first curriculum, which is a sequential curriculum, we start training our networks to perform the simplest possible task, which is the n=2 task. So the network needs to only remember the last two digits every moment of time. Then after the network learned this, we gradually increase the memory requirement of the task to n=3, n=4, and so on. And we could show that this kind of gradual increase of task difficulty is very helpful for RNNs to learn relatively long memory tasks. However, these networks suffer from a problem known as catastrophic forgetting, meaning that a network that is now trained to perform the task for n=20 can no longer perform the task for n=5, despite being trained for it throughout the curriculum. So they forget the things that they learned before. To solve this problem, we came up with a second type of curriculum, which we call the compositional curriculum. This curriculum also starts from exactly the same point. So we train the network to perform the task for n=2, but then we add a new readout head for n=3 in the next step, and then train it to perform the task for n=2 and 3 at the same time. And we just continue this as we go on by adding new readout heads. So in this way, we still have this gradual increase of the task difficulty, but we also bring in the compositionality. And this way, we force the network not to forget the things that they were learning previously. So let's see if there is a difference between the performance of these two types of networks. Surprisingly, we saw that the compositional curriculum, which has a more complicated loss function, is actually faster in training networks than the sequential curriculum. So within the same number of training epochs, the compositional networks would actually reach a higher end compared to the sequential networks. At the same time, the compositional curriculum finds more robust solutions. So after the training is done, if we start perturbing the parameters of the network, it could be the weights or the time constants that are learned, there will be a less reduction in the accuracy of the compositional networks compared to the sequential networks. So it overall shows that for whatever reason, this compositional curriculum is a better curriculum because it is faster, but it also finds more robust solutions. So the question is that what mechanisms inside these networks would create these differences in the performance? And of course, we're going to go back and look at the timescales in this case. So let's let's first look at the time constant parameter that we were training for each individual neuron in our network. We want to see how this time constant is changing throughout training as we are increasing the memory requirement of the task. You see that after some stage, so after we reach a task that requires a relatively long memory, then the sequential networks start adapting their time constant parameter to the memory requirement of the task. So they need this time constant to increase in order to be able to do a longer memory task. However, for compositional networks, in this case, you see a reduction in the time constant, and it looks like that it wants to try to reach the value that we put for the input update timescale, so how long each input digit was presented to the network. We can show this more explicitly by fixing the time constants of all the neurons in our network to a specific value, and then only optimizing the recurrent connectivity. And you see for the compositional networks, if I fix the tau parameter of all neurons to t this input update timescale, they would do as much as good as we were training them. So they actually don't need to change this parameter at all if we just give them the right value. However, the sequential networks do need training of this parameter and adapting it to the task memory requirements in order to perform the task. So now let's also look at the effective timescale. So this was a timescale that we measured from the activity autocorrelations. And what we expected from the beginning, this effective timescale increases in both networks as the memory requirements increases. So we expected larger memory, larger timescales in the network. But the interesting thing comes out when we compare these two timescales together. So for the sequential curriculum, we had an increase both in the time constant and in the effective timescale, which means that at least part of the increase in the effective timescale comes from the increase in the time constant parameter. However, in the compositional network, the only way that these networks could increase their effective timescale is through recurrent connectivity, because they would either decrease it or keep it constant, which suggests that the compositional networks are actually using recurrent interactions in the network to develop adaptive timescales that are relevant for the memory requirement of the task. So then in the second part, we saw that depending on type of curriculum we use for training our RNNs, they might actually come up with completely different mechanisms for solving very similar tasks. For example, in this case, they could either use biophysical properties or recurrent interactions. And depending on the type of mechanisms that they choose, then they might have also different computational properties. For example, training speed and finding better solutions was the property of the compositional curriculum. So now let's put both of these two parts of the talk together. In the first part, we saw that in primate visual cortex, adaptivity of timescales may arise from recurrent network interactions. And in the second part, we saw that the same mechanism, so using recurrent network interactions, is actually a useful thing for learning tasks that require adaptive timescale, which suggests that maybe like if we want to understand how adaptive behavior is generated in our brains, the crosstalk between like neuroscience and machine learning and AI could give us better answers if we put all things together. And maybe we would be able to find somewhat similar solutions across like both domains. But I would like to end the talk by taking a step back and trying to put things in a more broader context, which is the same slide as you saw yesterday, but maybe now it makes more sense after you saw what I already explained. So during this talk, I showed you that we potentially can find some relationship between behavioral timescales or computational timescales, for example, reaction times or the timescale of the memory and information integration with the timescales of neural activity fluctuations. And we can use recurrent neural networks, be it mechanistic like classical models or modern task-optimized models, to find potential mechanisms to explain how this relationship is built. But I think what is missing from this picture is the timescales of the environment. Because if you think about, for instance, the evolution, most likely our brains evolved to have these different timescales because we need to survive in environments that were very dynamic. Also, in our everyday life, we need to learn things that happens across different timescales. So the combination of evolution, development, and learning might actually help us better understand how these different timescales in the behavior and in brain are created. So this is something we are trying to explore during my postdoc using reinforcement learning models. And maybe the hope is that at the end of the day, if we combine both these mechanistic models and kind of more cognitive reinforcement learning models, we might be able to understand how, like in a broader context, different timescales of changes of environment would relate to different timescales of our behavior and brain activity. With that, I would like to thank my PhD and postdoc advisors, all my amazing experimental and computational collaborators, and most importantly, thanks again to all of you for being here today, and I'm happy to take questions.